Morning class. Today we will discuss about bioavailability. What is bioavailability? The rate and extent of drug reaching systemic circulation is termed as bioavailability. I'll explain you in simple terms. Whenever you take a drug, it must reach to your blood circulation. So the rate by which it's reaching your blood circulation and the extent and amount of drug reaching your systemic circulation is termed as bioavailability. So this bioavailability depends on three basic factors. What are those? Number one, pharmaceutical factors. Number two, patient related factors. And number three, route of administration. Pharmaceutical factors and patient-related factors we have already discussed while studying about solubility, while studying about the factors affecting absorption. So what are the pharmaceutical factors, which can also be termed as physicochemical properties which affects bioavailability? So number one, solubility of the drug. So the greater the solubility, the better will be the bioavailability. Next, dissolution rate of the drug. The greater the dissolution rate, the better the dissolution rate, the better would be the bioavailability. Next, polymorphism of the drug, amorphism of the drug, and pseudo-polymorphic nature of the drug. These are basically physicochemical properties which not only affects bioavailability but even affects solubility, even affects the absorption and many other factors. Next, stereochemical nature of the drug also affects the bioavailability. And next four factors which I am going to discuss is very important, and they are reoccurring in each and every kinetic parameter. They are particle size of the drug, lipophilic nature of the drug, pH partition hypothesis, and degree of ionization. These four factors are common in many kinetic parameters, also affects the bioavailability. So solubility, dissolution rate, particle size, effective surface area, polymorphism, amorphism, pseudopolymorphism, stereochemical nature of the drug, lipophilicity of the drug, pK, pH partition hypothesis, and degree of ionization. These are pharmaceutical factors which affects the bioavailability. Next, coming to patient related factors. Not only physicochemical nature, but the situation of the patient related factors also affect the bioavailability. What are those? Number one, age of the patient is very important, which affects the bioavailability. Next, gastric emptying time. This is the key factor. This is the time taken for your gastric content to get emptied into your intestines. So this time depends on your previous disease history, present disease condition, your food habits, your environment in which you are living, and many other parameters. Next, the pH of your GI tract. Next important factor is disease state, present disease state and previous disease state, and your medication history, your GI content, and your pre-systemic metabolism. In GI content, presence of drug in your GI tract, presence of food in the GI tract, presence of fluid in your GI tract, and normal GI content will affect the bioavailability. Next, pre-systemic metabolism, lumen enzymes, gut wall enzymes, bacterial enzymes, and hepatic enzymes will affect the bioavailability. These are the patient-related factors. Next, the other route of administration. So we have different route of administration. We are studying it from the first year. We have oral route of administration, we have intravenous route of administration, we have nasal route of administration, and many more. So when we compare all these route of administrations, every route of administration doesn't have same bioavailability. What do I mean? Every route of administration doesn't allow your drug to reach to blood in the same time. So parenteral route of administration among all available route of administrations will have 100% bioavailability, because in intravenous route you are directly injecting drug into your blood. So parenteral route is having 100% bioavailability when compared to oral, rectal, and topical. So when we see in sequence, first better bioavailability, 100% bioavailability will be the parenterals, then comes oral, then comes rectal, then comes topical. So these are the factors which affect the bioavailability: pharmaceutical factors, patient related factors, and route of administration. Next, what is the objective of bioavailability studies? In coming classes, we even discuss what is bioequivalence and what is the importance of bioavailability studies. So what are the basic objectives? To obtain evidence of therapeutic utility during development stages of the drug is the basic objective of the bioavailability studies, because we want to obtain the evidence that this particular drug is showing therapeutic activity not after releasing it to the market but during the development study. So that is the one of the objective of the bioavailability studies. Next, determining patient related factors, understanding possible interactions with drugs and efficiency of drug efficiency and even the absorption. This is the second objective of bioavailability studies. Next is a very important objective, controlling quality of drug product during the development to determine influence of excipients, influence of processing factors, influence of storage conditions, stability of the drug and drug absorption. All these parameters can be controlled, can be governed by understanding the bioavailability of the drug and chemical component. So bioavailability study is very important. Next, comparative studies between bioavailability of drug substances, existing drug substance and the new drug substance. So these are the basic objectives of bioavailability studies. What are those? I'll repeat: to obtain the evidence of therapeutic utility during stages of development, to determine the patient related factors, understanding the possible interactions with drug, efficiency of the drug and absorption of the drug, controlling quality of the drug products and determining the influence and the processing factors during the storage, stability and drug absorption, and comparative studies between bioavailability of drug substances. Next, what is absolute bioavailability and what is relative bioavailability? Now we have studied what is bioavailability. Next we will study what is absolute bioavailability, then what is relative bioavailability, then what is bioequivalence, then what is therapeutic equivalence, and then pharmaceutical equivalence. Absolute bioavailability is comparison of availability of drug when administered orally with comparison of bioavailability of the drug when administered intravenously. This comparison is called as absolute bioavailability because availability of drug when given intravenously is 100%. So you are comparing the 100% with the oral route of administration. This is absolute bioavailability. Then what is relative bioavailability? Comparison of availability of drug when given orally by oral standard of the same drug. So this is relative bioavailability. In absolute bioavailability, we are comparing the bioavailability or availability of a drug in the systemic circulation with the oral and IV. In relative bioavailability, we are comparing with the standard oral with the oral administration. So these are the definitions of absolute bioavailability and relative bioavailability. Then what is bioequivalence studies? So this is comparative study between two or more drugs, relative comparative study of two or more drugs and of identical dosage forms to know whether their bioavailability is equal or not. So it is identical bioavailability between two or more drugs, identifying the identical bioavailability between two or more drugs with identical dosage form is bioequivalence. This is nothing but comparison of bioavailability of an existing drug and a new drug. Comparison of bioavailability of the same drug of different dosage forms. Comparison of bioavailability of same drug and same dosage form but two different marketed formulations. This is bioequivalence. So it is equivalencing the bioavailability of two or more drugs. Next, what is therapeutic equivalence? This is comparison of two or more drug products with the same pharmacological effect which can control the disease at the same extent. So this is therapeutic equivalence. So comparison of two or more drugs, comparison of bioavailability of two or more drugs is bioequivalence. Comparison of pharmacological effect and therapeutic response and the extent of controlling the disease of two drugs is therapeutic equivalence. Then what is pharmaceutical equivalence? Comparison of two or more drugs and the drugs which are identical in the strength, quality, purity, content uniformity, disintegration and dissolution characteristics is pharmaceutical equivalence. Then what is chemical equivalence? Drug product containing the same label chemical substance as an active ingredient in the same amount is chemical equivalence. Students, we have studied today definition of bioavailability, relative bioavailability, absolute bioavailability, therapeutic equivalence, bioequivalence, pharmaceutical equivalence and chemical equivalence. So next, how to measure this bioavailability? So we have studied bioavailability, we have studied the factors affecting bioavailability, pharmaceutical factors, patient-related factors, route of administration. Now we will try to understand how to measure the bioavailability, how we are going to measure the rate and extent of drug reaching your blood, reaching your systemic circulation. So basically there are two methods. One method is pharmacokinetic method and the second method is pharmacodynamic method. Pharmacokinetic method is an indirect method and pharmacodynamic method is a direct method. And you know, in pharmacokinetics you have all absorption, distribution, metabolism and elimination parameters. So pharmacokinetic profile, which means you have all these characteristics, all these kinetic characteristics. Pharmacokinetic profile of drug reflects therapeutic effectiveness of the drug. So this is the key of this method. So there are two methods, indirect method, pharmacokinetic methods involved for measuring of bioavailability. One is plasma level time studies. Number two is urinary excretion studies, or urinary excretion method. In plasma level time studies, what are you going to do? You're going to take the drug and you're going to give it to the patient, maybe orally or IVly, whatever the route of administration. Then you're going to check Cmax, concentration maximum, Tmax, you're going to check half-life, you're going to check volume of distribution, VD, you're going to check rate of elimination, or you're going to check area under the curve. These parameters will give you the idea of bioavailability in the studies. Next, in urinary excretion rate method, you will understand the cumulative amount of drug excreted in the urine, that is changed drug excreted in the urine. So this will give you the indirect idea or measurement of bioavailability. So this is the first method, pharmacokinetic methods. Next is pharmacodynamic method. Pharmacodynamic method is also mentioned as it's a direct method. Here you have two methods. One is acute pharmaceutical response, pharmacological response, and second one is therapeutic response. Dear students, you will study about therapeutic drug monitoring and clinical pharmacokinetics and therapeutic drug monitoring when you will go in PharmD 5th year. You have another complete other subject dedicated to therapeutic drug monitoring over there. So pharmacodynamic method, in pharmacodynamic method we have two methods. One is acute pharmacological response, that is after giving the drug, after giving the dose, you will measure dose response curve. What this is what you are doing in pharmacology practical labs. So you will give the dose and you will check the response. What kind of response? Changes in ECG, maybe changes in EEG, maybe dilation of the pupil diameter. Next, it may be increase in the body temperature of an experimental animal or the human being or something else like that. So acute pharmacological response after giving the dose. With changing the doses, we'll check the changing the pharmacological response. So this is the direct method, because directly you're giving the drug and you are checking the dose response. So dose response curve and pharmacological effect versus time. These are the techniques of studying the acute pharmacological response under pharmacodynamic method of measuring bioavailability. Next, therapeutic response. In this method, you will directly check at up to what extent the disease is being controlled and how the disease is being treated and what is the therapeutic response of the drug. So these are the two basic methods of understanding of measuring the bioavailability. At last, before going into the bioequivalence studies, I would like to bring your notice to the BCS classification, biopharmaceutical classification system of the drug. We as a pharmaceutic students, we are going to study these bioavailability bioequivalence studies because we want these studies to help us in understanding the drug responses, in understanding they will help in us in clinical trials before releasing the drug into the market and all other things, while even formulation development of different dosage forms. So in order to improve the bioavailability, we'll see in next classes, there are different techniques of enhancing bioavailability, maybe by enhancing the solubility, maybe by enhancing the permeation, all those things. So to understand that thing, all the drugs are classified depending on the biopharmaceutical classification into 4 classes. Class 1, the drugs which have the high solubility and high permeability. Class 2 are the drugs which have the low solubility and higher permeability. Class 3 of the drugs which have the high solubility and low permeability. And the class 4 drugs which have low solubility and low permeability. So depending on the solubility of the drug and depending on the permeability of the drug, bioavailability will vary. So we are classifying all available drugs or all available chemicals in the market into 4 classes. Class 1 have the high solubility and permeability, which means the bioavailability is good because solubility is good, so its absorption is good, and permeation is good, so it is easily crossing the barriers, easily crossing the cell wall, and easily reaching the receptors. So bioavailability is also excellent. So no need to enhance the bioavailability by changing certain techniques. The second class of the drug has a lower solubility but better permeability. So in this class of drugs, we need to work out on the solubility to enhance the bioavailability. So if we succeed in enhancing the solubility of class 2 drugs, then we can improve the bioavailability. So this is an ideal candidate for increasing the bioavailability of choosing for the development of dosage forms or development of certain drugs. Class 3 have the better solubility but low permeability. Here we will try to enhance the permeability of drug by using certain permeation enhancers, or by changing certain techniques, by using certain pharmaceutical techniques, formulation techniques. We are going to enhance the permeability. So even this is an ideal characteristic, ideal drug, because you need to work either on solubility or you need to work either on permeability. So 2 classes, class 2 and class 3, are ideal for dosage form developments because you need to work either on solubility or you need to work either on permeability. Class 4 drugs are the worst drugs because you have low solubility and even the low permeability. So if you want to choose this drug for the dosage form development, then you need to work on both solubility and permeability, which is going to be tough. So ideal characteristics are either you work on solubility or you work on the permeability. So if you are going to select a drug for the development, so either take the class 2 drugs or take the class 3 drugs. So this is the BCS classification of drugs. Students, in coming classes, we will discuss about the bioequivalence studies. We will discuss about the study designs. We will discuss about the protocol of bioequivalence studies, and we will discuss about the in vitro and in vivo correlations in bioavailability and bioequivalence studies. Thank you for watching this video, and continue subscribe my channel and like this video and comment please to make me understand what improvements I can do to make you understand. Thank you.